Okay, class, we will get started or kind of finishing up part 1 of chapter 6. So first we want to understand coronary arteries. So we have a right and a left coronary artery. We can see that they branch off the aorta and they are on the exterior surface of the heart. So even though the heart pumps blood to the entire body, it has its own blood supply to fuel the heart muscle. A question I asked the students or the class is, why are these arteries on the exterior of the heart? So what the answer kind of is for that is it keeps the blood separate, so it ensures that as much blood goes through the cardiac cycle as possible, but it also allows the heart to receive its own blood supply. And then it also ensures that these arteries are not compressed during contraction. Um, let's talk about what these arteries kind of use to communicate. So anastomosis is the intercommunication between these arteries. So what the purpose of this is, it ensures blood flow even if one of these arteries becomes blocked. So atherosclerosis, which is plaque buildup, is very common in the aging population. It's caused by smoking, poor diet, lack of exercise, alcohol, high blood pressure. And in all of those instances, atherosclerosis plaque can build up and it can block blood flow from getting to the heart muscle itself. So what happens is anastomosis. So if a fully or partially blocked artery occurs, this cross communication between adjacent channels or arteries serves as a backup route for blood flow if one artery is blocked or compromised. So this still ensures that we get as much blood to the heart muscle, which is the myocardium, as possible. So let's talk more about that cardiac cycle. So we already know the path of blood flow to or through the heart. Next we want to understand is a term called systole and diastole. So we are all adults. I'm sure we've all had our blood pressure taken and we know what these terms mean. But let's go into a little bit more detail about each of these. So we know it's a sequential series of contractions. We know that one side of the heart contracts at the exact same time the other side contracts. So with this, we have systole, which is the contraction phase. So this is when the blood is pumped out of the heart. And then we have diastole, which is when the chambers relax. This allows them to fill with blood. So these next steps right here kind of show what would happen. The atria are filled. Atria are filled, they undergo diastole. They then pass through the valve. The atrias contract and now blood fills into the ventricle. So as the atrias undergo systole, which is the contraction, the ventricles will then undergo diastole. They will relax and they will fill. And then the ventricles will contract. There's a brief pause, 1/10 of a second is the amount of time that is lapsed before they contract after they have already filled. So this sequential series of contraction and relaxation of chambers enables the heart in a single effective pump. So one cycle of this circuit is a heartbeat. So this basically, the systole, diastole, when you put all of that together with the chambers contracting and relaxing, that's where one heartbeat occurs, or the lub-dub that you hear when you listen to a heartbeat. Let's talk about what controls this process. So we have intrinsic control and we have extrinsic control. So let's discuss intrinsic control first. So intrinsic control, you know the word intrinsic just means to come from within. So this is control within the heart and it's called autorhythmicity. So autorhythmicity, the ability of cardiac muscle to initiate impulse for contraction at regular intervals. So what controls this? The SA node, so our sinoatrial node. So control within the heart by this node without the central nervous system initiating the impulses. We'll talk about extrinsic control, which does have to do with central nervous system control, but not this SA node or the intrinsic control that we'll discuss here. So what is the SA node? We see it in the picture right here. It's at the very top of the right atria. specialized cells that have the ability to spontaneously produce an action potential. So the pacemaker of cardiac contraction. So it initiates when the chambers relax and when they fill. So this node controls everything. So why is this important? Why is this node, why does it have to set this pace? 2 reasons. It makes sure the chambers are filled first. So it makes sure that all the chambers are filled before there's the impulse for the chambers to contract. Also make sure there is no backflow. So basically it just guarantees that the chambers will contract in a specific sequence so blood moves in the correct direction. The discharge rate of the SA node is about 60 to 80 times per minute. And that sounds familiar, right? Kind of sounds like a heartbeat or a heart rate. And that's exactly what it is. The SA node determines your heart rate. The AV node is second. And let's go back and look. Where's the AV node? It's located at the bottom of the the right atria. So the AV node. This node is also specialized nervous tissue. It's in the lower portion of the right atria. So it spreads from the SA node to the AV node next. What's the purpose of this node here? It delays that impulse. We looked at a few slides ago. The impulse is delayed by 1/10 of a second. This is basically, why is this important? Basically, it's because so the atria can contract before the ventricles. It allows the ventricles to relax and fill with blood from the atria. Then what happens next? The impulse is spread through the right ventricles, passing through something called an AV bundle, and then the right and left bundle branches. Then it will pass to the Purkinje fibers. So we see that these Purkinje fibers allow the impulse to the ventricles to contract in a synchronous manner. It basically guarantees that blood is being pumped effectively. Purkinje fibers also use as little energy as possible. So it makes just the heart really efficient. So when we have everything synchronous, we have a more efficient heart rate. The next slide will show the pathway, and then we'll watch a little video. So our SA node up here has control over both these top atria. And so basically the SA node basically sends the discharge or determines the discharge rate. And so it sends the signal to the AV node. We said the AV node will delay the impulse by 1/10 of a second before sending that signal down through kind of like the septum, which is where in between the right and left ventricle, so in through this cardiac muscle wall. So this is basically the myocardium right here. So sending this signal down, and then you see it travels back up. So the Purkinje fibers exist kind of right along the ventricular wall, basically allowing the ventricles to contract together. So let's watch our quick video.
Any comments, Doug?
Yeah.
After
Only pay for what you need with Liberty Mutual.
Doug, where are Limo's pants?
You'll have to ask Lena.
This animation explains how a cardiac cycle is produced by electrical impulses in the heart. The cardiac cycle starts when an electrical impulse or action potential is generated by the sinoatrial node. The electrical impulse travels along specialized conducting fibers in the walls of the atria, stimulating the atria to contract and pump blood into the ventricles. In the normal heart, impulse travels very rapidly. As a result, all the muscle cells in the atria contract together, producing a coordinated pumping action. The impulse also travels across the right atrium to the atrioventricular node, where it is delayed slightly to allow the ventricles to relax and fill with blood before they contract. The impulse travels along the bundle of His, where the conducting pathways divide in two, forming the left and right bundle branches, which lie in the interventricular septum. From the bundle branches, the impulse spreads into the inner walls of the ventricles via specialized conducting fibers, called Purkinje fibers. Finally, the impulse travels up the outer walls of the ventricles towards the atria. The rapid and controlled spread of the electrical impulse through the ventricles stimulates the muscle cells in the ventricles, to contract together and pump blood efficiently to the lungs and body. As you will see, during atrial fibrillation, the electrical activity of the atria becomes disorganized. As a result, the pumping function of the atria is lost, and the rhythm and efficiency of the entire heart are disturbed.
So pretty neat. What's next? So I think it just goes over this one more time for studying purposes for y'all. So starting with the SA node, we even have another picture that kind of shows the heart or the structures of the heart, so you can really see it. So SA node will contract the right and left atria, so we can follow where that signal goes. Then arriving at our green little AV node. So the lower portion delays that impulse just to make sure that these ventricles or the atria contract kind of appropriately. Then the signal travels then to the AV bundle, which is located kind of, right here. And then the right and left bundle branches, which are at the bottom, eventually to the Purkinje fibers. So this is just the path of intrinsic control, and you do need to be familiar with this pathway. This slide here looks at that same intrinsic control, but now looks at an EKG or an ECG. It shows you the P wave, QRS complex, the T wave, and the U wave. XFIS 3305 does not really spend time going into this. I know that testing and prescription will cover this in a lot of detail. So I'm not really going to spend any time with it, but it's interesting. So I just kind of wanted to show you that the SA node, basically when the atria contract, that's our P wave. So that's our atrial depolarization. We see then the signal will spread to the AV node, which will then allow those ventricles to contract, and that's our QRS complex. And then the relaxation of the ventricles, the recovery wave, that's our T wave. So that's really all I'm going to go into about the ECG. You can also just kind of look at normal heart rate, fast, slow, irregular. The video at the top is a good video just to kind of look at. Again, there might be a commercial. Let's see.
Good morning, sunshine. This is going to be a great workout. We're going to go ahead and get...
But here we
The
go.
electrocardiogram, or ECG, is a recording of the heart's electrical activity via leads placed at standardized points on the subject's body. This animation shows how the different ECG waves are produced during a cardiac cycle. The P wave is produced as the electrical impulse travels through the atria, causing them to contract. The P wave corresponds to atrial depolarization. The impulse then travels through the atrioventricular node, producing a flat section on the ECG, known as the PR segment. The PR segment is flat because no current is flowing through the cardiac muscle cells. As the impulse travels from the AV node to the bundle of His and along the upper part of the septum, the Q wave is produced. The passage of the electrical signal through the ventricles produces a large spike called the QRS complex. The QRS complex is much larger than the P wave because the ventricles are much larger than the atria and so produce more electrical activity. Most of the QRS complex represents depolarization of the left ventricle, which has much greater mass than the right. The impulse remains unchanged for a moment as the ventricles remain depolarized. This gives rise to another flat section called the ST segment. As the impulse fades away and the ventricles repolarize, the T wave is formed. Note, this animation shows the ECG recorded from a lead placed at position V6, one of the standard electrode positions to the left of the heart. Leads at other positions detect the electrical activity of the heart from a different point of view, and therefore produce differently shaped ECGs.
Alright, so let's continue. Now talking about extrinsic control. So just remember, intrinsic control is autorhythmicity. It is the control from within the heart. So extrinsic is external to the heart. So we have to talk about parasympathetic nerve fibers and sympathetic. So first, let's look at where this control comes from. And it comes from the medulla oblongata. Think back to chapter 5. We talked about what structures of the brain have control over our breathing rate, our blood pressure, heart rate. We said it was the brainstem. The brainstem is kind of broken into multiple different parts. There's the pons, the midbrain, and the medulla oblongata. And so that's what we're looking at here, is the medulla or the medulla, either way you want to say it. All right, so we're going to talk about parasympathetic nerve kind of stimulation first. So parasympathetic nerve fibers are also called vagus fibers, and we see that right here. If we follow this little green line, we see it activates our SA node and the AV node. So when these fibers are activated, they're going to release acetylcholine. Think again, back to chapter 5, we said that parasympathetic nervous system or parasympathetic stimulation, it releases acetylcholine. What does it do? That's the neurotransmitter. It's the chemical that will decrease the SA node and the AV node. So I normally ask the class, what else will decrease if the SA node and AV node activity slows down? And that's heart rate and blood pressure and breathing rate. There's something else that we can discuss called parasympathetic tone. We'll discuss more about this in the next little lecture video, but parasympathetic tone is increased vagus nerve fiber activity at rest. So this is higher in athletes, so especially aerobic. Aerobic athletes have higher or greater or increased parasympathetic tone. And so what this is, it's a lower heart rate at rest. It's just this increased parasympathetic sensitivity. So now let's look at sympathetic nerve fibers. We see down here at the very bottom. Now we've gone kind of further down the spinal cord. So kind of more so in the spinal cord, this is where the sympathetic cardiac nerve fibers come in. They release norepinephrine, and we already know that as well, because in chapter 5 we said that sympathetic nervous system will release norepinephrine and epinephrine. We'll talk about epinephrine on the next slide. So with norepinephrine, what happens? When these fibers are activated, they're going to increase SA node and AV node activity. And when the activity of those nodes increases, the cardiac cycle also increases. And in that case, the myocardial contractile force will be greater. So what that means is just the contractile strength of the heart. So when we have an increase in norepinephrine and the AV and SA node, what else will increase? Again, it's going to be heart rate and then volume of blood pumped. And so why is there a greater volume of blood pumped? Because of the increased myocardial contractile force. The heart's going to contract harder, which will eject more blood. So yeah, that's kind of the basics of our extrinsic control of sympathetic nerve fiber. But let's look at the role of the adrenal glands. So this is also extrinsic control. So we have sympathetic nervous system will stimulate our adrenal glands. These glands sit on top of our kidneys. We'll talk about in chapter 8, we have an adrenal medulla and an adrenal cortex. So with this, these are kind of our fight gland, where they release epinephrine and norepinephrine. We kind of already know the role of epinephrine to increase heart rate. So this picture here shows something called the thoracic splenic nerve. What that is, it's basically relating to the internal organs kind of around our abdomen. So these nerves arise from T9 and L1 of the spinal cord and innervate the adrenal gland sitting on top of the kidney. So that's kind of really all that shows. But what we want to understand is how does the adrenal gland releasing epinephrine, how does that influence our heart rate? We see up here a word called bradycardia versus tachycardia. Again, I always ask everybody, what is the difference between these two? So with bradycardia, it's going to be a heart rate less than 60 beats per minute. And then tachycardia is greater than 100 beats per minute. So the 60 to 100 range, that's considered normal. So 60 to 100 beat per minute, normal sinus rhythm kind of throughout the day, because we know it changes throughout the day with exercise, rest. But yeah, so less than 60 beats per minute for Brady, greater than 100 beats per minute for Tacky. All right, so let's continue. So just kind of summing up what our extrinsic control does, it has to be a balance, and our body works on a balance between multiple systems. So in this case, it's a balance between intrinsic and extrinsic. So what we want to look at next with extrinsic, and I have them kind of blocked over here, is something called a chemoreceptor. We may have already mentioned it previously, but chemoreceptors are kind of clusters of cells that detect changes in the composition of our arterial blood flow. So mainly going to the head and brain for the aortic arch chemoreceptor, and then the carotid arch chemoreceptor detects blood going to the head, to the brain. The aortic arch chemoreceptor detects blood going to the body. So what these receptors do is they're basically specialized sensory receptor cells which transduce or convert a chemical substance into a biological signal. The signal is basically an afferent sensory signal that is sent back to the brainstem or the medulla to respond to changes that are detected by these receptors. And so we'll look at on the next slide the role of each one of these. So chemoreceptors, they monitor chemicals. So goal is to maintain homeostasis for these receptors. So maintaining homeostasis in the cardiorespiratory system by monitoring chemical levels. So what chemicals are we talking about? CO2, carbon dioxide, is produced by our working muscles. So chemoreceptors will detect if blood is high in CO2. H+, which you know as hydrogen ions. So hydrogen ions are also detected as a chemical. And then partial pressure of oxygen, or PO2. We'll discuss more about that in chapter 7, but all that means is a low level of oxygen or a high level of CO2. So when there's an increase in CO2 and hydrogen and a decrease in partial pressure, that usually means, or maybe one example would be, we probably just got done exercising. So our muscles put off CO2, there's an increase in acidity or hydrogen ions, and there's a decrease in partial pressure of oxygen because our muscles are using that oxygen. So we have an increased sympathetic signal to increase heart rate. So all of that would occur during exercise. Baroreceptors, which were not shown on that picture, but are also receptors that we have throughout our body, especially in our heart, and they detect pressure. So baroreceptors monitor pressure. So if we have an increase in blood pressure, so our body detects that, and it's going to decrease our heart rate and contractility to try to lower that pressure. And then same, or kind of vice versa, if we have a decreased blood pressure, we're going to have an increased heart rate and contractility to increase that pressure back up to normal. So this slide here was just kind of added to show you the kind of effect that kind of takes control or happens to each kind of one of these systems. You read at the top, it's just major factors that increase our heart rate and force of contraction. So you don't have to really know much about this slide. We've already talked about proprioceptors in chapter 4, so increased rate of firing during exercise. We just talked about chemoreceptors and baroreceptors, how they detect pressure or chemicals. Catecholamines, we know that epinephrine and norepinephrine will increase our heart rate. Calcium, potassium, sodium, temperature, all those will be regulated and increase with exercise, which again will increase our heart rate. So I think that's all we've got for part 1 of chapter 6.